Welcome, cell biology students, to the third presentation. This presentation is going to focus on cells and energy and how cells can use energy to perform work. What we're looking at here on the title slide is an enzyme. And an enzyme is a protein. Okay? It's a polypeptide chain. It's a string of amino acids that have assembled into some higher order conformation, and in this case here it's actually 4 proteins that have assembled again into a holoenzyme, or a larger type of biochemical machine that can be used to perform work. And it's a very specific type of reaction that each type of enzyme can help to catalyze or promote. And all of these kind of topics relate to energy, so let's take a look at that coming up. We'll start by looking at the use of energy by cells. We'll talk about free energy and catalysis, and that's important to talk about when we think about whether some sort of a reaction is favorable or whether it is spontaneous. And if a cell needs to perform some sort of a reaction that isn't favorable, how does it go about doing that? And we'll also look at activated carriers, which are also part of this discussion because they act as kind of intermediates to be able to drive unfavorable reactions. These are molecules that contain groups and high energy linkages, but we'll look at that at the end of the discussion. So how do we get large, structured, organized molecules from simple building blocks? It takes energy to do that. And then once we have these large macromolecules, in some cases they perform jobs. They facilitate biochemical reactions, which also takes energy. So how do we do these types of things? First, some kind of basic background. We can think about a series of events within a cell where a cell has some sort of a molecule, right? And this is arbitrary, so this is molecule A in this case here. Let's say the cell requires something different. It needs to modify molecule A into something else that it's deficient in, or something else that it's required to synthesize. How does it start making changes to molecules? These are performed by enzymes. Right? We looked at an enzyme in the title slide, but enzymes are proteins, and they're a type of protein that performs work, and their job is very specific and very dependent on the enzyme's conformation. In this case here, enzyme 1 takes molecule A and tweaks it into molecule B, and that's the job it does, and that's the only job it can do. In this case here, we call this a substrate and a product. So it's something that the enzyme physically grabs onto and then helps to promote the modification into. So this is what it binds to and what it changes that thing into. All right, but let's say the cell doesn't need this kind of intermediate molecule. It needs some sort of kind of end product. How does it continue to perform biochemical tweaks and modifications? It requires other enzymes. Okay, so if enzyme 2 is present, it can take molecule B and just molecule B and change it into molecule C. Right? So it takes this substrate and makes this product. Okay. Enzyme 3 can come in and do another specific job, and so forth. So in this way, a cell can take some sort of a molecule and perform a series of biochemical reactions upon it that result ultimately in something that it needs. And the purpose of this slide is to demonstrate that this requires a team of enzymes, and a team of enzymes working together to perform their discrete jobs. And together we can call this a biochemical cascade, which is a term I really like, but it's a cascade of events, almost like a cascading waterfall or something like that. Right, so just with terminology again, very basic, we have an enzyme. Enzymes have shape and conformation. Oftentimes that shape and conformation includes a pocket or an active site, which can physically match whatever molecule it's going to bind onto and modify. It's substrate. So it can bind to a substrate, and there's a physical kind of lock and key association between the two. There's an interface and there's surface area in between the enzyme and its substrate. Okay? And that's reinforced by some of the bonds that we talked about previously. Electrostatic, hydrogen bonds, maybe there's hydrophobic pockets or something like that. But there's a physical match, right, that's oftentimes reinforced by bonds and so forth. So the enzyme here can catalyze a reaction or promote a certain type of reaction that changes the substrate into the product. And then ultimately the product is released, and that's how we can generate a pool of product from a pool of substrate. A couple things to think about here is that with this enzyme, it's performing this kind of cyclic type of activity, binding substrate and creating product. But one of the defining characteristics of this cycle is that the enzyme isn't changed at this point within each cycle. So every time it gets back to kind of being open and free and ready to perform its job, it is consistently in that same shape. It's not changed. What's changed here is the substrate to product. So we can look at a kind of a real-world example of what that cartoon was showing a simple example of. And one case is with steroid synthesis, and I'm going to walk you through this, and you aren't responsible for memorizing the enzymes that we'll look at or the intermediate products that we'll look at. I show you this because I think it's a useful example to look at what we've learned with that simplified model. So steroids are a type of hormone, and they're all modified from cholesterol. Ultimately, this is the initial kind of building block. So cholesterol can be acquired in the diet. It integrates into cell membranes and modifies the chemical properties of the membranes. But also importantly, it can be modified into different types of steroid hormones which have very different effects in the body. So there's a set of glands called the adrenal glands that sit on top of the kidneys. And the adrenal glands have a region called the adrenal cortex. And within the adrenal cortex, there are three discrete kind of subregions or zones. And one of those zones within the adrenal glands is called the zona reticularis. Now within the zona reticularis, there are cells that contain the specific enzymes to step by step modify cholesterol ultimately into some different types of sex hormones. And one shown here is a type of androgen. You can even look at these molecules and see that they're bearing some similarity. Right? You can see some of these conserved kind of rings. But the enzymes are responsible for doing the step-by-step modifications of cholesterol to get to this end product here. Within the adrenal cortex, there's another zone that contains different cells, right, called the zona glomerulosa. So these are different cells that contain, right, some different enzymes. So these enzymes are performing their own specific jobs, taking one substrate and converting it to a product, taking that product and converting it to a different product. Right, the series of events that we looked at with that simple cartoon. And as a result, you get cholesterol forming aldosterone, which is, has some similar structures, which is fascinating, but at the organismal level and the influence that it has in cells has a very different role. Okay, so let's look at a third example here, the zona fasciculata. Okay, so this is the cortical, most outside or superficial region of this gland, the adrenal gland, and it contains different cells with different enzymes. Right? So because you have these different enzymes, they're taking molecules and modifying them differently until you get to cortisol and corticosterone. So these are what we call glucocorticoid hormones. But again, there are similarities there that can be appreciated just looking at the molecular structure, but there are also very important differences in the molecular structure. That means that within the body at the cellular level, they're going to have very different roles. Okay. So this is a busy slide, and there's a lot going on, but I want to show you a real example of the way that these things can kind of play out, and how you can take one sort of molecule, a substrate, and convert it into all sorts of different things, depending on what enzymes are present. And if we look at the reality of the situation in all its glory, right? This is a illustration of all of the pathways mapped out within one cell. So these are the biochemical cascades where you have substrates being converted into products, but it really highlights just the really astonishing reality of the complexity of all of these different biochemical cascades all put together. And that's one way it's useful to start simple and to show a simple cartoon. Then kind of look at how that can be used in a real world context with the steroid hormones, and then also to really understand how complicated it really is. But they're all consistent in that they're, for the most part, just enzymes taking one molecule and changing them to another. Okay. And to really take a step back, again, and think now kind of globally about the way enzymes are modifying molecules and breaking them up and building them down and performing changes, we can think about cells requiring some sort of an initial food or energy source. But this food or energy source needs to be broken down so that the building blocks can be used to then synthesize something different. And they need to be broken down because that releases energy. So here we have food molecules shown being broken down, and when these food molecules are broken down and the bonds are broken, there's heat that's lost. And once these building blocks are kind of freed up, they can be used to synthesize something that's required at the cellular level. Okay? And some of this energy that's released can be converted into intermediate forms of useful energy that can be used to drive biochemical reactions. I suppose one way to kind of think about this would be, let's say you eat a jelly donut or something like that. There's lots of carbs in a jelly donut, but those aren't particularly useful at the cellular level. But if those carbs are broken down into glucose, right? Glucose is really useful. That's simple sugar at the cellular level. And that glucose can be broken down further to generate ATP. And ATP contains useful forms of energy that can be used to perform work and to synthesize molecules and drive biochemical reactions. Or let's say you were to eat protein, like in the form of soybeans or something like that. Soybean protein isn't going to be particularly useful for the proteins that you need, let's say in your skeletal muscle. Right? Your skeletal muscle is going to contain different types of proteins, like actin and myosin and other types of proteins. So how do you get the building blocks to make actin and myosin from the proteins found in some sort of a plant tissue? Well, those proteins need to be broken down into the amino acids so that they can be assembled into the proteins that are required in a particular cellular context. Okay, so we have food sources, which are important for building blocks, so cells can make the things that they need. We have food molecules that can be broken down because in that process, energy is released and can be converted into useful forms of energy, like the synthesis of ATP, which can be used to drive biochemical reactions. So we can break things down and build things up, and we call these catabolic pathways and anabolic pathways. So catabolic is to break down, anabolic is to build up. If you want a way to remember that, you can think cats break things. So I have a cat here tipping over a vase. Catabolic is to break down. I'm going to show a couple examples of energy use at the cellular level. We've looked at how enzymes can take molecules and change them from one to another. They can tweak their molecular structure. That requires energy. But there's also some processes which at the cellular level are clear examples of something that requires energy because it looks like work. So one example is with something like a flagellum, which is that whip-like type of structure that some cells have in humans. They're only found on sperm cells. But this is clearly an energetic process. You can see the motion. Right? And if you look at the structure of a flagellum, like a cross-section here, this is a transmission electron micrograph, so this is electron microscopy. If you take a cross-section of a flagellum, you can see that it's highly organized. So not only does the motion of a flagellum require energy to drive changes in protein shapes, which is really driving the shape of that structure, but also the organization of the flagellum and the synthesis of the components that were used to build the flagellum. Those processes require energy. This organization requires energy. Why does it require energy? Well, that kind of opens up this discussion of the second law of thermodynamics, which is in any closed system, like the universe or any system within it that's closed, these systems tend to disorder. So without any sort of input of energy, they tend towards chaos. The example that a lot of books use, including yours, is this cute example of a nice tidy room. And without any input of energy, it's going to tend towards more chaos or more disorder. And then to get it back into shape requires an input of energy. So we're going to use that example, that kind of cute analogy at the cellular level coming up here. But if it's not energetically favorable to increase organization or order, then it's not energetically favorable to build cells or to build the molecules that cells require to do the things that they need to do. So that's not energetically favorable. So how is that possible? How can you kind of disobey the second law of thermodynamics? It's made possible because there's the release of heat energy from cells. And heat is super chaotic, right? It's basically increased activity and vibrations of molecules increasing to their chaos. So this is how we reconcile the organization you see at the cellular level, which is something that doesn't disobey the second law of thermodynamics. So shown here is a cell, and it exists in an environment, and there's molecules, intracellular and extracellular. And as the cells synthesize macromolecules, like proteins or DNA or other types of polymers, that process is also generating heat. So these biochemical reactions generate heat, right? And heat increases disorder in that system as molecules begin to migrate around more chaotically. So heat is bringing chaos to the system, which can offset the organization that's entailed by the building of macromolecules. Right? So even though it looks like the system is creating order, right? More broadly, there's increased disorder. So that's shown here, right, with the heat that's lost. That's how we can get from food molecules broken down into building blocks and assemble and increase some of the order that's suggested by cells. is that within this process, there's heat that's lost, and heat is brings a lot of disorder to the table. So it brings a lot of chaos. So ultimately, where is a lot of the energy being fed into kind of the cellular world? Where is the energy coming from? Where are a lot of these building blocks coming from? Where's the kind of primary source of energy? A lot of that's coming from photosynthesis, or from photons from the sun. So some organisms have special approaches to being able to use photons to basically synthesize organic molecules. Okay. So here we have an example of sunlight striking some sort of photosynthetic organism. At the cellular level, there are molecules that can absorb some of those photons and orchestrate a response with the input of these photons to split water and generate what are called activated carriers of energy. ATP and NADPH are examples, but these are kind of intermediate energy carriers. They're molecules that can be used to later on down the line drive biochemical reactions, and in this case here with stage 2 of photosynthesis, they can be used to synthesize and manufacture sugars. Okay. So stage 1 requires the input of photons that can modify some of the molecules within specialized cellular structures, together with the input of water. This stage here results in the manufacture of intermediate high energy carriers that can be used to manufacture sugars. Okay. Part of stage 1 results in the production of oxygen, right? And in stage 2, there's the fixing of carbon dioxide and the integration of carbon dioxide, particularly the carbon, into large macromolecules. So now we can take that previous slide and just combine it, the two stages of photosynthesis, stage 1 and stage 2, right? Requires sunlight, of course, requires water fed in, right? And releases some oxygen, as well as the input of carbon dioxide to synthesize, for the photosynthetic organism's sake, organic molecules and sugars. Now there are other organisms that can use these sugars and organic molecules as their food source and break them down for building blocks, and also break them down to generate their own high energy bond carrying kind of intermediate molecules like ATP. Right? In this process here, the breakdown of these molecules releases carbon dioxide, and the synthesis of energy via cellular respiration requires oxygen. Okay, produces water. Okay, so this is how the input into the system very initially requires photons. So organisms that have the unique ability to take those photons and the energy within those photons to synthesize sugars, these sugars and other organic molecules can be then fed into the broader kind of global cellular system, so that cells that don't necessarily have that unique ability do have access to the molecular bonds and the molecular building blocks to do the things that they need to do. Okay, so cells obtain energy by the breakdown of organic molecules, the oxidation of organic molecules, which leads us to a discussion briefly about oxidation and reduction. And this is involving the movement of electrons. So let's say we have atom 1 and atom 2 here, and they're different atoms, and they form a covalent bond. We talked about this with hydrogen bonds in the last discussion, that hydrogen bonds result when you have a hydrogen that's covalently bound to another atom, and that other atom has a tighter grasp on the electrons, where the electrons aren't equally shared within that molecule. The electrons tend to spend more time over by the more electronegative atom. That's typically either oxygen or nitrogen. So here, right, these two atoms come together. Let's call this one hydrogen and this one oxygen. The electrons spend more time over by the oxygen. So as a result, because electrons are negatively charged, that part of the molecule has a more negative charge. It has a partial negative charge. But we call it reduced. Right? So the negative electrons have a negative charge, and they've reduced this part of the molecule. Whenever you have reduction, you also have the opposite. Whatever the counterpart is there, we say it's become oxidized. It has a partial positive charge. One way to perhaps help you remember that is oil rig. Oxidation is loss of charge, right? And reduction is gain. Right? O-I-L-R-I-G, oil rig. And we can also look at that in kind of a real-world example here and think about a particular atom within a molecule and whether it's getting oxidized or reduced. So shown here is a very simple organic molecule, methane. It has a carbon right here at the core, and this carbon is sharing pairs of electrons with 1, 2, 3, 4 hydrogens. It has a covalent bond with 4 different hydrogens. If we add an oxygen into this molecule, now it has an hydroxyl group off to the side here, so now this is methanol. We call this carbon now oxidized. There's an oxidation that occurs here because the electrons are now, to a greater extent, attracted to the oxygen. Oxygen's electronegative. So the electrons are effectively spending less time over by the carbon. Right? So the oxygen's getting reduced, and the carbon is getting oxidized. We can add another covalent bond here with the oxygen here, and that's drawing electrons to a greater extent away from the carbon. So now this carbon is even more oxidized. Right? The electrons are spending less time over by the carbon because they're attracted to the oxygen. Right? Throw in another oxygen, right? It's increasing the oxidation of the carbon. So with this series of changes to the molecule, the carbon is getting increasingly oxidized. Right? Or conversely, right, we can take carbon dioxide and have it undergo a subsequent series of molecular changes, and with it the reduction of the number of oxygens within that molecule means that the electrons are no longer attracted to the oxygen, and they're sharing more of their time, spending more of their time with the carbon, so the carbon's getting reduced. Right? So oxidation, increasing the oxygens, increasing the attraction of electrons to the oxygens, or more importantly, reducing the time that they're spending with the carbons, is oxidation. And then the opposite is reduction. So with part 2 of the discussion, we'll be looking at free energy and catalysis. We'll bring the discussion back to enzymes and what enzymes are doing. Again, enzymes are proteins, and they perform some very specific job within the cell. And you can think about them as taking some sort of a molecule, some sort of a substrate, and modifying it, or breaking a bond, or perhaps helping to contribute to the synthesis or adding of two molecules together. So they're performing some sort of a very specific biochemical job. And the way that they promote the progression of these biochemical reactions is by making them more likely to occur. Even some reactions that would happen spontaneously or would move a molecule from one state into a more energetically favorable state may happen, but just may not happen very quickly. So a cell needs to increase the rate at which these reactions are occurring and the likelihood that they'll occur. So they make these reactions more likely to occur by reducing energy barriers or barriers to a reaction. So let's say there's a bunch of molecule Y and the cell requires X. Right? An enzyme can step in and make this biochemical reaction, right, taking these reactants and producing these products, more likely to occur. So in this case here, Y would be the substrate, right, and then X would be the product. So the representation most books will use is to look at some particular reactant Y, right? The cell requires X. This is even a favorable reaction, so that's shown by having less total energy. So this is a favorable reaction, but there's a barrier to it occurring. So this is the activation energy. Some references will use the analogy as a campfire or something. So wood burning is favorable, but it doesn't generally just happen, right? You need to add something, some sort of a match or something along those lines to get the process kind of rolling. That's the barrier. For enzymatic reactions, this is what we're talking about here. There's a barrier to the conversion of Y to X. So with the addition of an enzyme, right, this is more likely to occur. There's less barrier. Another way to think about this is if you have, shown here in green is the number of a particular type of molecule, right? So let's say this is Y, and so this is the substrate. Y is going to exist in a variety of different levels of energy as molecules vibrate and crash into each other. They exist in different energetic states. Down here, you're not going to get any of these converting from Y to X. But a very small fraction of that population of molecules exists in a very high, just for a brief moment, state of energy. And if it's sufficient, it can convert from Y to X. And some of these will have enough energy to have surpassed that barrier, to have exceeded that barrier. If you add an enzyme, that barrier is reduced. So even molecules of lower energy can undergo that chemical reaction. So a greater proportion of molecules will be able to be converted Y to X. Okay, so it's just another way to think about it. Right? Y to X, right? The cell needs X. Even if it's favorable, there's a barrier to that biochemical reaction. You add an enzyme and it lowers that barrier. Okay, here's another illustration the book uses. So there's a couple different ways to think about this. They're all kind of saying the same thing. Right, so you have these molecules that are existing in various different energy states, but without an enzyme, very few can get over the hump to be converted, right, Y to X. But an enzyme is going to decrease that barrier, right? So there'll be greater numbers of molecules being converted into X, which is great because that's what the cell needs. So it may also be that a particular molecule, Y, has a potential to undergo all sorts of different types of chemical modifications. In this case here, this is arbitrary, but there are 4 different options of chemical reactions that could occur, but 3 of them are useless, so they wouldn't do the cell any good. what an enzyme can do is selectively decrease the activation energy for a certain type of reaction. So it's not only promoting the changes of one molecule to another, but it's also directing. So it's kind of encouraging one type of change over another. So here we're looking at a particular type of molecule, and there's a whole world of molecules that could be converted into, but a cell with the appropriate kind of complement of enzymes can drive a particular type of biochemical cascade. Many of these could be relevant in other cells, and other enzymes might drive the change of this molecule into something else. But in one particular cell, the presence of a particular kind of cohort of enzymes will drive one kind of predominant biochemical cascade. And we looked at this to start, right? That's one of the first examples I showed you was with the steroid hormones. And in that case, it was cholesterol. But depending on what enzymes are present, it's going to reduce the energy of activation for particular types of reactions, and also make the likelihood that an entire cascade will lead ultimately to one desired product here, one of these particular types of steroid hormones. One thing we want to talk about with reactions and kind of understanding whether a reaction is likely to occur or not is by talking about free energy. So free energy is going to determine whether a reaction can occur. It can be thought of as useful energy. And in favorable reactions, it's going to be negative. So here the free energy we call G. The change in free energy is the delta G, so delta for difference. And there's going to be a difference before and after as a molecule changes from one substrate to one product. If that delta G between those two different states is negative, then it's favorable. Even if it's spontaneous, that doesn't mean it happens immediately. Right. So it may be that an enzyme helps to move some sort of a spontaneous reaction along, just instead of this reaction taking minutes, months, or years, right, it takes fractions of a second, which is the timelines that cells need to be working on. Why is the free energy negative between these two states with a spontaneous or kind of a favorable reaction? It's because there's the release of heat. So there's the release of heat energy, which is lost and not useful energy. So that means the free useful energy before and after is negative because some of that energy has been lost as heat. An unfavorable reaction is one with a positive G. Okay, you might think of this as being biosynthesis, right? So taking amino acids and combining them into a polypeptide chain. There's something along those lines. That's increasing order, and that's not favorable. So how do we get unfavorable reactions to occur? Because certainly a cell needs to build macromolecules, needs to increase order, needs to synthesize proteins and nucleic acids and sometimes long chains of carbohydrates and things along those lines. How do these unfavorable reactions move forward? Well, fortunately, they can be coupled. So you can take some sort of a reaction that's unfavorable, so it has a positive delta G. Maybe this is the building of a protein or something like that, increasing order. And you can couple it to something that's negative. And if the negative reaction, the negative delta G, is larger, you can add these up, right? So that now the net is favorable. So one example of this would be a reaction where glucose is combined or a bond is formed with fructose. So this is increasing organization as the two molecules are added together. The two monosaccharides formed into a disaccharide. So it's not energetically favorable. And here we call this the standard free energy. So it has a superscript naught here. And this representation here assumes some kind of ideal conditions and reactant concentrations so that we can talk about different types of reactions in kind of an equivalent way. But note that it's a positive value here, which is not energetically favorable. So this won't spontaneously occur. But it's a reaction that needs to occur. So how is a cell going to be able to promote that? Well, it can couple it to a favorable reaction. So a great example of a favorable reaction is the hydrolysis of adenosine triphosphate, right, to adenosine diphosphate, where that third phosphate is removed. It's favorable, right? So here the standard free energy is a negative value. And not only is it negative, but it's greater than the positive value here. So if these two are coupled, this more favorable reaction can be used to drive a unfavorable reaction, right? But it's less unfavorable than this is favorable. So the net is still negative. And the value here is negative 1.8 kilocalories per mole. Here, the mechanism is that the phosphate from adenosine triphosphate was removed and tacked on to an intermediate form here where glucose has a phosphate tacked onto it, preceding the condensation here where you get the formation of the disaccharide. So the take-home message here is that unfavorable reactions, many of which are necessary at the cellular level, can move forward if they're coupled to very favorable reactions, have a bigger negative standard free energy. We talk about standard free energy because that allows us to compare different types of reactions without thinking about concentrations, because concentrations are going to affect the delta G. If there's a lot more substrate, it's going to push the reaction forward. If there's a lot of reactant, right, the reaction isn't going to be as favorable. Right? So concentrations with reactants and products influence the likelihood that a reaction will proceed. If there's already a lot of product, it's not going to be as favorable. So that's shown here, also with another kind of cartoon context of something a cell has, X, something a cell needs, Z. This substrate can be turned into this product, which then becomes a substrate for this product here, and the reactions are catalyzed, as shown by these arrows here. And also with these arrows, the size of the arrow demonstrates how favorable a reaction is. So in this case here, the conversion from Y to Z is very favorable, as it's shown with a larger arrow moving forward. But this is much less favorable, right? So let's take a look at these, each of them independently. Right, so here we have a reaction which is really not that favorable, right? Y is even being converted to X in this particular context right here. But the cell needs to take this unfavorable reaction and couple it to something very favorable. So in this case here, right, the reaction is very favorable, and most of the Y is converted to Z. And this means that if you add all of these together, X to Y, then Y to Z, because Z is kind of really siphoning or dragging and converting all of that Y molecule here, it's really dropping down the abundance of Y within this context. And because Y becomes very unavailable or very sparse, or there isn't very much of it, and the concentration drops way down, right? Another way to think about it is whenever there's a Y present, the cell very quickly converts it to an X, so there's very little Y just kind of available at any one moment in time. That's driving the movement of X to Y, right? So it's acting like a siphon. It's reducing the abundance of Y, which helps to promote this reaction right here. And so reactions are dependent on the concentration of the substrates and the products. So the equilibrium constant is one way we think about how much of a particular substrate or product there's going to be, dependent on how favorable a particular reaction is. So the equilibrium constant is shown here as the rate at which you take a molecule A and a molecule B within a cell, the amount that these two will associate, the favorability of these two molecules forming a bond and forming the product here, which is A and B. So there is a constant. There's a value, depending on the reaction, that can say, okay, so at a moment in time we have A and B at equilibrium, right? Step back and let the process kind of proceed forward as it naturally would. At the end of the day, how much A and B are you going to have combined? This is the association rate. But there's also a natural frequency at which this bond will be broken, right? So there's going to be a formation value, but also a dissociation value. So there are also rates that this molecule A and B, if we want to stick with the previous example with a disaccharide, there's going to be a rate at which that disaccharide will break apart, right? Will dissociate. Okay. And with the two of these together, the association rate and the dissociation rate, we can get an equilibrium constant, right? What is the value, or what is the concentration that you'll have of substrates and reactants if the reaction is allowed to proceed until an equilibrium is formed, right? You'll get the formation of the bond in some cases, you'll get the dissociation of the bond in some cases, and if it's just allowed to play out, then there's going to be a concentration of these two different pools of reactants and products, which is related to the equilibrium constant. All right, so activated carrier molecules and biosynthesis. We've already looked at one example, maybe the best example, is ATP. ATP is called an activated carrier molecule. And these are molecules, and there's more than just ATP, that are used to help to drive unfavorable reactions. So in this case here we have food molecules, right, which are broken down, and that creates heat and that creates chaos, and that's a favorable reaction, so that'll have some sort of a negative free energy value. And this is the breakdown. I said this is catabolism. Those breakdown reactions can be used to generate intermediates, or these activated carrier molecules that come in different types, but they harbor high energy bonds or have other chemical properties that can then be used to drive energetically unfavorable reactions. Right? So perhaps this is an anabolic reaction, like the combination of the monosaccharides into a disaccharide, like we saw with some of the previous examples. So ATP is a great example. In this case here, the group that's carried in a high energy linkage is phosphate. Right, so ATP, adenosine triphosphate, that third phosphate and that second phosphate share kind of a high energy bond or a linkage, and if that's broken, adenosine triphosphate into adenosine diphosphate, that releases energy that can be used to drive other unfavorable reactions. There's also some energy between ADP and AMP, but it's not as much, and usually when we're talking about ATP driving chemical reactions, we're talking about a cleavage of that third bond. NADH, NADPH, FADH2, These are also great examples of activated carriers, and we'll talk about these in more detail when we talk about like photosynthesis and the citric acid cycle and things along those lines, cellular respiration. But to also make note that there's other types of molecules that can be generated that also contain these high energy linkages. So ATP is the most widely used one, and the one we kind of talk about the most. Here's adenosine, right, triphosphate. There's an adenine, there's a sugar group here, a ribose, and then 1, 2, 3 phosphates. So it's definitely favorable, right? We saw that before with the negative 7.3, I believe it was, is the standard free energy with the conversion of adenosine triphosphate to adenosine diphosphate. So that was used to build the disaccharide in that example that we looked at. And in that case, the phosphate was covalently bound to kind of a glucose intermediate. So now you need to make more ATP, right? So if the energy has been pulled out of ATP and you're down to ADP, at some point the cell's going to have to replenish the amount of ATP. And how is a cell going to replenish ATP? I could do that through sunlight, if it's the type of organism that has that capacity. Or it could do it by breaking down food molecules like glucose, for instance, and taking bonds from those molecules and using them to increase the amount of ATP that's available. So ATP, also when low, another trick that can be used in some cases is for something called creatine phosphate. It's basically a molecule of creatine that's attached to a phosphate. That phosphate can be added to ADP to form more ATP. So it's kind of a quick, acute way to increase the availability of ATP, right? Grabbing phosphates from creatine phosphate. When ATP is high, right, and that's kind of the nature of energy and the reality of a cell's existence is that resources are going to kind of ebb and flow. So depending on context, energy levels can be high or low. And when energy levels are high, that's great, but it's also a good opportunity to do things like generate more creatine phosphate so that it's ready to go when energy levels are low to quickly generate more ATP. Right? So when there's lots of ADP, the cell's going to need ATP. So where's it going to get the phosphate to make ATP? From creatine phosphate. Okay. Conversely, if there's a lot of ATP, it's probably a good opportunity for the cell to make creatine phosphate for quick renewal or quick replenishment of pools of ATP. So energy stored in ATP is oftentimes used to join two molecules together, like we saw with the formation of that disaccharide, where initially there's usually an activation step. So ATP transfers a phosphate to some sort of a molecule to form the high energy intermediate, right? And we did see that before with glucose, right? So here we've got the arbitrary molecule is A, but molecule A is now tacked to a phosphate group, right? And then that phosphate group is pulled off, right? Which opens up the opportunity for a condensation reaction, right? For the formation of that bond or that larger molecule, right? That unfavorable piece of the reaction. As a result, a phosphate is lost, right? And because it's a condensation reaction, water was used. Okay, so the net result here is, right, A becomes activated so that it can be combined to B. Okay, so A is activated here, and then it can be combined to B, and you get the formation of a larger molecule. A real-world example would be glutamic acid. So this molecule here changed into glutamine, right? Initially there's the activation step, So a phosphate is pulled off of ATP. It's tacked here to this high energy intermediate so that an amine group can be added down here. So there's something else that's sent in to replace the phosphate, whereas attached to the molecule in the high energy intermediate. NADH and NADPH are also activated carrier molecules. They carry electrons and they're involved in redox reactions. So if you look at this particular case here, there's a molecule A, and within its molecular structure it has a hydrogen and 2 electrons. Those can be pulled off and attached to NADP positive. So this positively charged molecule here picks up the hydrogen and the 2 electrons. That's called a hydride ion. And it picks up those electrons and their negative charge, right? So it's become reduced. And it carries this hydrogen along with the 2 electrons that it can then deliver to another molecule here and reduce it. Okay? Remember, when there's a reduction, there's also an oxidation. These are redox reactions. So the molecule in this cycle is then oxidized and prepared to pick up another hydride ion. So it's a way to transfer a hydrogen and 2 electrons from one molecule to another and participate in these redox reactions. So, brief overview, we looked at the energy used by cells. We thought about energy in terms of chaos and how a closed system tends towards disorder and chaos, and that's favorable. But cells are very much about organization and the synthesis of large macromolecules and doing biochemical reactions that aren't necessarily favorable. So we thought about how free energy can be used to tie favorable reactions to unfavorable reactions, and how enzymes work to lower the energy of activation for specific reactions to occur, and in series with a cascade that can be used to take one molecule and convert it into a wide range of other molecules depending on context. And then we looked a little bit at activated carriers and how those can be used to drive unfavorable reactions. We'll look at all of those in more detail as we move forward, depending on context. So this is a little bit of kind of an overview of energy and some of the basic concepts. All right, as always, let me know if you have questions. Hope you're doing well. Take care.